Got that? Awesome. All right, so today we're going to be talking about greenhouse gas emissions from reservoirs. And we have two researchers who study that very topic. The first one, we have Professor Alex Forrest from UC Davis, who's going to talk about how to quantify greenhouse gas emissions from Mediterranean climate reservoirs. And then we have Dr. Elisabet Perez from UC San Diego, who's going to talk about a kind of surprising emissions pathway from ultra oligotrophic reservoirs for methane. So at this point, I mean, climate change doesn't really need an explanation or any introduction in California. It's really beating us over the head. Everybody probably remembers the Lake Oroville spillway disaster that happened in 2017. And in the time since, I mean, it would take your kid to maybe complete their hydrology degree at UC Davis or something. 4 years later, here's the reservoir. And these are things that we have to deal with as lake managers. These impacts of climate change are just obvious. While I was putting together this presentation, I learned that of the 20 largest fires in California history, 17 have occurred in the last 20 years. And I've heard a lot of people use the term fire season recently, which I don't really like that. That's not really a thing that I grew up hearing, but it's just become a reality of living here, living in the state. And all you have to do is go outside in the summer and take a deep breath in to notice it. So climate change, we often think about the impacts that climate change is going to have on our water supply and our water quality. It's not just a water supply issue. So climate change can actually cause reservoirs to stratify for longer and remain anoxic for longer. You could have more release of metals and nutrients from bottom sediments that are going to both affect how treatable water quality or water is for source water. And it's also going to cause things like harmful algal blooms, which we've seen increasing in the recent years and months. A recent study showed that 80% of California native fishes are in decline. And at the current rate, about 45% are likely to be extinct in the next 50 years. And so as reservoir managers, we're always thinking about the effects that climate change are having on our reservoirs. But few of us are thinking about the effects that our reservoirs are having on climate change. And some recent studies have shown that reservoirs and lakes are actually major sources of greenhouse gases to the environment. And the issue here is that when you release greenhouse gases, you have more warming that actually releases more greenhouse gases. And so you get stuck in this positive feedback cycle that degrades water quality, and also it just adds further uncertainty to our water supply. And so to talk about that today, we have two speakers. So the first is going to be Professor Alex Forrest. Alex is an associate professor of civil and environmental engineering at UC Davis and a past CALMS president. With a background in civil and environmental engineering, his research examines the influence that lake bed and seafloor features have on localized hydrodynamic flow. And Alex uses acoustic mapping techniques and environmental fluid mechanics to interpret the dynamics of a system. And so with that, I'll let Alex share his screen, and he'll give us a little discussion on reservoir greenhouse gas emissions.
Somewhere here. Is that the right view, Mark?
Yep, looking good.
Great. All right, so thank you, Mark, and welcome everybody. And today, like Mark mentioned, I'll be talking about greenhouse gas emissions, and particularly from some reservoirs in Valley Water. So this is actually a partnership between UC Davis and in civil engineering and Valley Water itself. So we entered into a collaborative research agreement last year. And so there's a large host of characters from both Valley Water side and our side that are involved in this. And so it's really been one of the silver linings for us coming through COVID is getting involved in this research. It's been interesting times and interesting results as well. So just to briefly introduce the problem of methane. We know, so this is a plot on the left of methane gas production. And for a while in the early 2000s, we thought we were sort of plateauing out. But then we started to rise back up again in terms of global methane production. So on the right are two plots. One is remote sensing of methane, based from 2003 to 2005, and then again from 2008 to 2010. And what the general consensus is a lot of this is coming about through climate change and heating of temperate and polar regions, and all of a sudden releasing more methane into the system. And part of these sources of methane that have become increasingly acknowledged are aquatic ecosystems. And so understanding lakes and reservoirs is one of the critical things that we need to do in order to understand the impacts on climate change. So just to sort of put this more into the context of reservoirs, because most of our systems in California are reservoirs. So here's a very typical example, or a schematic, where we have some sort of impounded water. So we have a dam that we have river inputs coming into the system periodically, and this is bringing organic material. It brings more organic material in to a reservoir that gets deposited on the bottom. And then during periods of anoxia, we then start converting this organic matter through anaerobic degradation into methane. So we start getting methane generation and carbon dioxide generation that comes into the water column in our hypolimnion that can either, well, then gets diffused into the system. So we have a diffusive flux that tends to be relatively constant. And then we get emulative or bubbling flux that's coming off the bottom. So that's sort of that bubbly schematic or bubbly cartoon on the right, trying to show sort of these episodic releases of methane. And those are the two main mechanisms I'll be talking about today, because that's been the focus of our work to date. That said, there's also N2O that can be formed, that's another greenhouse gas, although it's not as large a contributor to the problem. And so that's one of the reasons why we haven't focused on that to date, although it is a potential element to this problem as well. The other element that I'm not going to talk about, but is sort of recognized, is that methane release and degassing happens a lot in rivers. And so as you release this water downstream, if it's rich in methane and dissolved gases, when it goes out into the river, you have a potential additional source to be thinking about when you're doing the overall carbon balance of a reservoir system, both in the reservoir, but also in the outlet downstream. So we know that methane emissions are coming out of aquatic water bodies. So the y-axis here is on a logarithmic scale, where lakes and reservoirs, so the second and third colored dots on the left, are some of the biggest contributors to methane production globally. And this isn't just a function of latitude, but also trophic status. So how rich the systems are in nutrients. So we know, and I'll show you a slide in a second, that eutrophic systems that have high nutrient content have much higher flux rate. So as an example of this, Clear Lake in north of San Francisco will have a much higher potential methane release than, say, something like Lake Tahoe that's ultra-oligotrophic. So knowing the trophic state of your system and knowing how that varies is part of the problem. So I also spoke about the three main compounds of methane, carbon dioxide, and nitrous oxide. These all play a role in greenhouse gas emissions, particularly as you're moving into the high trophic states. So, for example, here in this box plot, this is in terms of milligrams CO2 equivalents, with the eutrophic system on the left has the highest potential contribution of methane as compared to carbon dioxide and nitrous oxide. Now, for this presentation, I'll be showing methane as methane rather than CO2 equivalents, even though 1 milligram of CO2 is worth roughly 25 times that of, sorry, one milligram of CH4 is worth about 25 times that of CO2. So trying to understand this is a big piece. And then also trying to understand the drivers. So there's been a number of studies trying to understand globally what's driving the formation of methane in these systems. And there's been some large comprehensive studies. With that said, these studies tend to be focused on both boreal lakes as well as tropical lakes. And the area that tends to be ignored are temperate or Mediterranean climate lakes like we have here in California. Now, the little work that has been done does show that air and water temperatures are major drivers. And so that's part of what we need to focus on, particularly in future climate change scenarios. The other part of this, and so these are various elements in terms of area, maximum depth, DOC, chlorophyll, etc. The black squares are reservoirs, whereas the open circles are lakes, as taken as much larger inventory. And the strongest correlation that's being shown so far is the presence of chlorophyll. So now the amount of chlorophyll content. So now it's not just a matter of temperature and bottom temperature, but it's also the amount of chlorophyll content that can potentially be contributing to your methane production. And this is, again, extremely important in California, where we know over the last several years that we are increasingly getting large algal blooms during the summer and harmful algal blooms during the summer in many of our lakes and reservoirs around the state. So trying to understand that and trying to understand that correlation is important. So I mentioned at the beginning, we've been collaborating with Valley Water. So we set up this collaboration last year where we went out and we have a number of different test sites. So these are 3 reservoirs in the system. First of all, we have Stevens Creek, Chesbrough Reservoir, and then Uvis Reservoir. They are all roughly the same depth, although because it was such a dry year, the smallest of the 3, Stevens Creek, got quite shallow. But we're sort of talking maximum depth of about 10 meters, 10 to 12 meters depth. And impounded on one side, and then a flooded riverbed going up. And so as the water level drops, basically we lose ground at the inlet end of the lake. And then at each of these sites, you can see basically locations. So we have these little pinned locations where we go out and collect multiple samples. So at each of these sites, we collect triplicate samples, and then we've been doing surveys across all 3 lakes on a quarterly basis. And then at Uvis, which is this one here, at this location, right now down near the deepest end of the lake, we've been going back and doing 24-hour sampling every month to try to understand the seasonal variation. And here's what we see. And I'm going to show the CO2 results here, although we're going to focus the rest of the discussion on methane. So here on the methane side, in winter, so black and blue, February and March, when the study started, we basically had very little production of methane coming off the bottom. But then as time goes on, we start seeing an increase in methane where here in June and July, we have the largest peaks of methane being produced. Now, this is done by time. So what we've done is in a 24-hour sampling period, we try to go hourly and collect triplicate samples to get a single data point. And we do this with a team of undergraduate students that have been joining the graduate students involved in the project to try to capture this diel variation, to really understand the flux rate coming off the bottom. Because without understanding that, it'll be hard to quantify the greenhouse gas emission coming off these systems without understanding the overall seasonality. So basically, how do we compare summer to winter, but then also how do we compare the peak production at 3 in the afternoon with minimal production at 3 in the morning. Now, CO2 results do vary as well. It's a more complex story based on the productivity of the system. So this is one that we're still looking into because during some days of the year, these systems can be a net source and sometimes they can be a net sink. So trying to understand that, or net sink of CO2. So in order to understand that and how that balances the methane, the net source of methane, is something that we're trying to understand for this system. And the other question or challenge in this is that we're not just experiencing temporal variation, but also severe spatial variation. So I mentioned earlier that there were 3 locations across the lake, basically near the outlet, in the middle, and near the inlet. And for each system, we then went out and gathered in our quarterly sampling or seasonal sampling samples from each of those. And as you can see from this table, there is a high degree of variation just across those three samples. Plus, there's also a large degree of error in the standard deviation because the samples themselves do experience a lot of variation across the lake. So trying to understand the spatial variation and trying to understand the temporal variation is a key piece to understanding how these systems will respond into the future, and also what their overall carbon input is to a system, to the atmosphere. So what we do know is that the reservoir production, the methane production, is a significant contributor. Now, not as much as wetlands. So here's a table of wetlands, boreal lakes, peatlands, et cetera. The values that we see coming off of these lakes vary between, say, 10 and 20. So we're just below this table in terms of milligrams per meter square per day. But that's still significant, a significant contributor. And then also trying to understand the spatial variation across the system. So it will depend not just at the time of day, but where you're measuring across the system, across the lake or reservoir. The other thing is that CO2 also displays the same degree of variability. So if you were going out to design a field campaign to try to monitor this, you would really have to take all these different factors into account. So the other thing that we've been doing is trying to understand the primary drivers. This is some very initial data where we've been, there's a cross-sectional profile of the reservoir up on the top panel with the outlet at one end, the inlet on the other. And at certain locations where we've been going out and capturing gas samples, we've also been putting in a hydrolab sonde to get water quality parameters, such as water temperature, pH, ORP, and DO, and chlorophyll. And very initially, what we're seeing is the relationship between the dissolved oxygen, basically the presence of anoxia in the system, will contribute to methanogenesis, so that the methane production will go up. So this is something that we're still trying to understand and fully characterize all the different parameters. And some of the initial look that we've done, although we're still in the process of collecting more data here, is that the middle one here, the relationship between bottom water temperature and the diffusive flux rate, is the strongest correlation, although we're still hoping to refine that correlation based on gathering more data through the rest of the year. And then we're also seeing a weaker correlation, but still an existent one, between methane production and chlorophyll presence. So the other piece of this that we've been trying to capture is the idea of emulative flux, basically the bubbling methane that's coming off the bottom. So this was in collaboration with another group at UC Davis who had developed an open source methane sensor. And what we did with this is we built a small floating gas chamber like you see here on the upper left, and installed this open source piece of equipment inside to measure methane continuously. And what you can see is this slope of the line, and then a little jump, and then it slopes up, and then jump, and then slopes up. So that slope is how we're estimating the rate of the diffusive flux versus the jumps, these small little jumps, and the larger one that I have highlighted here. These are representing basically the bubbles coming into the chamber. So you get an ebullition event, it pushes methane into the chamber, and then it jumps up to the next level, and then it will start increasing again because of diffusive flux. And this is one of the big challenges with trying to map out where this ebullitive flux is happening, because it's so episodic and so spatially distributed. So this is the next stage of how do we quantify this, because ebullitive flux can be up to 30–40% of what your diffusive flux is, if not more. So trying to get an idea of the overall amount of methane coming from the system, you have to quantify both those pathways. And so one of the ways that we try to address this is using hydroacoustics. So this is based on the work of Anderson and Martinez from 2018. And they developed a new technique using a biosonics echo sounder. So this is 199 kilohertz echo sounder, essentially a fancy fish finder that they would carry around on a boat and then use that to sonify the bottom. And then they related the response, basically how hard the echo return was from the bottom to the amount of gas being stored. And they developed this relationship here with these black dots, or the black symbols are the data that was taken from the Anderson and Martinez paper. We've then gone in 2 different seasons and added our own samples, so the blue and the green, to help refine this result and try to come up with a universal relationship between the response in the sediment to the actual gas content. So we designed a new chamber, and the new chamber would capture gas being released from the bottom that we'd lower down to the bottom, capture gas inside, and compare that with the actual instrument that we had on board. So this is an example from Oroville Reservoir from this is November 1st last year. And Ruth Thurkel, one of my graduate students, she was out mapping the system, so mapped the system, and then has color coded it by the response of the hardness of the echo return, put it through this algorithm here, and then the output of this algorithm then gives you the methane content in the lake bed. Now so the bright colors, so down here at the deepest end of the lake, the yellow and the green, we'll just ignore the red stars for a second. The yellow and the green are showing basically higher gas volume stored in the sediment. And so that's an interesting way to look at being able to capture the variability across the system to be able to go out and map. She could basically map the reservoir like this in a single day, come back and process the data and come up with a map of where the gas content is. The other interesting thing that she saw is what she's colored with these red asterisks, which represent bubble plumes in the echograms. So basically looking at the echogram, which is basically a 2D curtain plot of basically what streams across on a fish finder, she's able to see and tag the geolocation of each time she saw a plume. Because when the students are out there in a small Zodiac or a small boat, they're sitting there on the water, and you can literally see spatially distributed around you. You can see small bubbles coming up in various plumes. And so this technique of being able to identify these plumes helps us understand where the emulative flux is happening versus higher gas storage. And there's a fairly good correlation between higher gas volume in the bottom along the thalweg of the reservoir versus the littoral sides, which don't have any evidence of these bubble plumes at all. But there's also seasonality to this. So this is an example from Stevens Creek. So on the left is late October. On the right is from mid or late February. And unfortunately, this particular reservoir was extremely low in water level this year. And so we weren't able to get up into this larger arm, the inlet arm, because there was simply no water. But in the deepest part of the reservoir, you can see here higher gas storage and then bubble plumes popping up across the bottom. Now, we still have higher gas storage in the winter, even though it's reduced from the summer. So there's a seasonality there that we're capturing with these hydroacoustics. But then we're also not seeing any of these bubble plumes come up. So that would also indicate that the diffusive flux plays a more constant role through the year than the emulative flux, which can have its own seasonality where it's turned on and off based on how much gas storage there is. And here's another example of trying to track this seasonal ebullition. So here we have UVIS, fall, winter, spring, summer. And where in the fall, late fall, when we really get a lot of peak in our production and gas storage from the perspective of using the hydroacoustics, we have a lot of sediment gas here that's contributing to the diffusive flux, but that's also being reworked in the water column. And then these bubble plumes coming up there, these then disappear in the winter, just like they do at Stevens Creek. But then in the spring, they start to reappear intermittently and then start to really appear in the summer when production gets turned on again. So understanding these variances and these subtleties between emulative flux and diffusive flux and their overall contribution is a critical piece. So some preliminary conclusions. And this study is still in its infancy, but what it is showing is that this subset of California reservoirs displays strong daily, high during the day and low at night, and seasonal, high in the summer and low in winter trends in methane production. The correlations exist between chlorophyll A and water temperature, leading to increased methane production in future climate scenarios. And what I mean by that is that we see these correlations between chlorophyll A and water temperature, and between that and methane production. And we also know that in future climate scenarios, we're likely going to get warmer in California, and we're also likely going to have more harmful algal blooms and chlorophyll production. And so this is all leading to a potential scenario where we'll have more methane production in the future. The hydroacoustic surveys suggest a spatial distribution of gas storage and provide methane to capture the bubble plume formation. And that's a pretty powerful tool that hasn't really been explored to date. Now, unfortunately, I haven't really spoke to this yet, But the initial study that we had set up was to look at the impacts of an HOS system at Stevens Creek. And that's why Stevens Creek was included in this study. But unfortunately, because of the low water levels, the HOS system was never actually turned on in this reservoir. So that's something to be explored going into the future. And I apologize for my voice, it seems to be cracking a little. So future work. So some of the open questions that's applicable here, but then also applicable across the entire state, is how do we scale greenhouse gas emissions across these systems in Valley Water and other systems around California? How does reservoir management affect greenhouse gas release? So in controlling levels, can we have an impact on what the greenhouse gas release is? How are levels of chlorophyll A related to the methane emissions? And what really is the relationship? Can we improve on our observational strategies? And in doing so, how much can we improve the accuracy of the flux measurements? Then how will these emissions change in future climate scenarios? And this is where we need to be doing modeling of future climate scenarios under various IPCC climate scenario into the future. And then finally, what role does nitrous oxide play in these systems? And is that potentially a significant contribution? So I won't take questions now. I'll leave it there and let Elisabet talk. And then I'll take questions at the end.
Awesome. Thanks, Alex, for a great talk. Yeah, like Alex was saying, if you have, you can just drop them into the chat and we'll be fielding those later at the end. Our next talk is going to be about a totally different flavor of methane production and emissions from lakes. And we have Ellie here to give that talk. You can go ahead and share your screen. Elisabet Perez is a postdoc at UC San Diego. Her current research focuses on understanding the role of microorganisms in the carbon cycling of leaf decomposition in rivers and streams of the Pacific Northwest. She received a PhD in environmental systems from UC Merced, where she studied the methane cycling dynamics of Sierra Nevada lakes and how environmental changes affect methane production and consumption in this ecosystem. So I'll turn it over to Ellie. Still there? I think she might have dropped off for a second. Okay, I just saw Ellie on, so she should probably just be popping right back on. In the meanwhile,
sorry, I pushed the wrong button and
then no worries.
closed the meeting. Just give me a sec to share the screen.
Oh, no problem.
Mark, you may have to make her a co-host again. It looks like that went away for some reason. Oh, there we go.
Cool, just did that.
Yes, thanks.
Cool, looks good.
Yeah, can you see my screen? Thanks for the patience. Yeah, so thanks for the introduction. Sorry that I dropped in the middle of it. And hello, everyone. I am going to be talking about my part of my PhD research that is now titled Co-occurrence of Paradoxical Methane Production Mechanisms in High Elevation Lakes. And because Alex gave a really good introduction about methane in lakes, I'm gonna skim a little bit in the first 2 slides of my presentation. We know that methane is like a very significant gas in global climate change, and it's increasing in the environment. And particularly for methane, microbial activities are very important, since microbes basically produce and consume the majority of the biological made methane in the environment. And as mentioned before, freshwater lakes are significant contributors to the methane budget. And they also have a big variation in the emission estimates, given this high temporal and spatial variability in different reservoirs and lakes. And so here, also Alex kind of like explained some of these dynamics, but I just want to focus in the parts that are going to be relevant to this talk, and that is focusing on the processes that happen in the sediments, which is the microbial process that produces methane is methanogenesis, as mentioned. And this process is inhibited by oxygen, which is why in general it happens in the sediment, in the sediments of the bottom of the lakes. This other microbial process called methane oxidation consumes a lot of the methane that is being produced by methanogens. Namely, it can be 30 to 99% of the methane produced. And as mentioned, the difference between the methane that's being produced and what is being consumed in the water column, it will also depend on some, however much this gets actually diffused into the atmosphere is gonna depend on some other physical parameters, such as wind speed or the surface layer of the lake. But overall, this is the main traditional view that we have of how diffusive emissions happen in the lakes. However, there is a significant component that we've been kind of overlooking over several decades, which is there have been consistent observations of methane being accumulated in the surface water of lakes and oceans. And since methanogenesis, the microbial process that produces methane, is inhibited by oxygen, this kind of it's a paradox that this is happening where it happened, whereas there's a lot of oxygen, since you wouldn't expect to have a process inhibited by oxygen in a high oxygenated area of a lake or an ocean. And that's why this common observation of this is called the methane paradox. And so we know from these observations of methane in high oxygenation areas that this happens, and it's been kind of noted in different studies over the decades, but not until like the last decade, people have been studying in the actual biological mechanisms that this might be happening. And this phenomenon, well, it's suggesting that there are other processes besides methanogenesis that are contributing to methane production in lakes. And even though it's been observed for a while, we're just kind of like starting to understand what other biological process can be contributing to methane production in lakes and in oceans. And so from now on, when I talk about paradoxical methane production, I basically mean any other process that produces methane that is not traditional methanogenesis. And so some of the few things that we do know how this might be happening in lakes is that it's possible that methanogenesis happens in oxygenated areas as long as it happens in micro-anoxic zones that are floating, for example, in the surface of lakes, which would be a case maybe if there's like a micro-anoxic zone in fecal pellets floating in the water or inside of the gastrointestinal tracts of organisms like fish or zooplankton. There's also been some observations of methane being produced on the surface of phytoplankton. So for this case, we're still seeing methanogenesis, but it's happening in zones where are devoid of oxygen within a larger oxygenated area. There's another prevailing idea mostly observed in the ocean so far, which is the methylphosphonate assimilation or methylphosphonate breakdown. And this happens when certain microorganisms are trying to access the phosphorus within this molecule. And during the breakdown of the carbon phosphorus bond releases methane as a byproduct. And this happens a lot in the oceans because they're very phosphorus depleted. So this is important also in the context of lakes that are oligotrophic, where there might be a major competition for phosphorus or other nutrients. And most recently, it was found that cyanobacteria can fix inorganic nitrogen into methane, but the pathway hasn't really been defined. The main thing that we know about this pathway is that it happens during photosynthesis and mainly by cyanobacteria. And some of the reasons why I wanted to study this methane paradox process in lakes was because it's kind of like a mysterious process that is still like, there's a lot of things to learn about it. We don't know exactly all the mechanisms or players involved in it. And we know that this could be an important source of methane to the atmosphere that we're not really accounting. And given the uncertainty that we have on these methane budgets, I think it's really important that we further study what's happening in these lakes, if there's these other processes that could be contributing to methane emissions. And the two major research questions that I wanted to ask, that I wanted to answer with this research was how widespread is methane production in the oxygenated water column of high elevation lakes, and what are the potential mechanisms of aerobic methane production in lakes as well. And the way that I went about to answer these questions is we selected 5 lakes in Yosemite National Park. So these are like oligotrophic lakes in high elevations. And these 5 different lakes had like a temperature and elevation gradient. And the idea of this was like that they would be characteristic for the many thousand lakes of lakes that are present in the Sierra Nevada. For future reference in the next slides, the names of the lakes are Lukens, Lower Cathedral, Upper Cathedral, Lower Gaylor, and Upper Gaylor Lake. And so I started a field campaign in these 5 lakes, and we mostly collected samples in the summer, fall of 2016 to 2018, every 2 to 2 weeks. We were kind of limited to the access of the lakes in the wintertime because of snowfall. And it had a field and laboratory component in which I collected samples in the lakes of the waters and bring them back to the lab for analysis and to perform this lake water incubations from these different lakes over the 3 years. And from these incubations, we measure methane and dissolved oxygen concentrations over time. And we use different tools to answer the research questions. Mainly, we extracted DNA and RNA to measure the metatranscriptomes, metagenomes, and stable isotopes from the gas to identify the potential pathways for aerobic methane production. And in conjunction to these, we quantify the functional genes that could be related to different methane production mechanisms. And in these experimental incubation experiments that I set up, we were mainly measuring methane accumulation or consumption over time. So like initially, we just wanted to see if this happened in the water of the lakes that we collected. And beyond that, then we use a couple of treatments that would hopefully help us explain the mechanisms of methane production in this incubation. So one was the effect of high light intensity or absence of light in the experiments. And the other one was the addition of a methanogenesis inhibitor for the different incubations. And so I'm gonna go ahead and start talking a little bit about the results. And so I mentioned the first research question was how widespread aerobic methane production in high elevate was, or this paradoxical methane production. And I guess in the short answer of this is like that we found active methane production in 37% of the experiments and methane consumption in 16% of the experiments. And this is shown in this graph over here, where you can see the methane production rate on the y-axis, and on the bottom, you can see the different incubation experiments that I ran over the 3 years. So it was a total of 19 incubations, and they're characterized here by the initial of the lake and the incubation experiment. So for example, this L1 experiment would be the experiment from the water collected in Lucan's Lake in July of 2016, while L2 would be water collected from Lucan's Lake, but like in August of 2016. And then so these are all the lakes, all different time points, and the different results that we found from them. And yeah, you can either see production or consumption, and then the few ones that are in gray, it means that we didn't see any statistically significant changes over time in methane concentrations. And you could see that from the previous graph, but this is emphasized a little more, which is like this methane concentrations varied across lakes and times of sampling, and also within replicates and time points. And we interpreted this as, we know that there's two processes at least that are happening in these lakes, which is methane is being produced and consumed. And for all of these that are unamended, there shouldn't be anything impeding either happening. And so both of them could be occurring at the same time, potentially. And this is something that it can be shown in this figure, where you can see the change in methane concentration over the incubation time. And one example of where we could observe that both production and consumption happen in the same incubation was for this bottom UC3 experiment, where you can see that methane is initially increasing, and by almost the end of the experiment, it kind of seems to be consumed almost completely. And this is something that we could capture, but it's possible like in certain time points that we just didn't sample, something similar happened where like methane went up or down. So this just kind of like, it seems like there was a lot of variability in the experiments. So the data that I showed before, it was for incubations that we didn't use any kind of amendments. It was just lake water incubations, and we observed what happened over time. But we also had incubations, and we used experimental treatments to try to find what are the potential other pathways that are happening in methane production. And in this figure that I'm showing right now, you can see the log response ratio on the y-axis and the different incubation experiments on the bottom. And I'm gonna talk about the different treatments a little bit, which is the top one is the BS treatment, is the one where we added the methanogenesis inhibitor. And so for this experiment, we were expecting to see a negative response ratio, which would mean that the methane concentrations were lower in the treatment compared to the control. But as you can see, this methanogenesis inhibitor didn't consistently inhibit methane in all the incubations, and even in some of them, it seemed like the treatment had a higher methane concentration compared to the control. We also had this dark incubation, in which we remove any light coming into the incubation bottles. And for this one, the reasoning for this treatment was, since light inhibit methane oxidation, we were trying to see if there would be increased methane oxidation with the light component. But we didn't see that in all of the cases, only in a few of them where we saw the negative response. But I guess the main also important point here is like the bottom one, where we like increase the high light intensity. And we were trying to see with this experiment, let's say that this methane production that is happening in the incubations, if it's related to cyanobacteria or photosynthesis related, increasing the light intensity would potentially have a positive effect in it. And this is actually something that we saw, but we had less incubations with this treatment. And a summary of what I just showed you is like, we had a higher variability responses similar to what we saw within the controls. But this could indicate that there's different mechanisms of paradoxical methane production in the experiments. And so we use different techniques and biochemical measures to try to figure out the actual pathway that could be producing methane in these oxygenated incubations. And so our first approach to answer this question was to analyze the DNA and RNA from the lake water of the incubations. And from this, we could basically get all the genomes of all the microbes that were inside of the inside of the water that we were incubating. And this could tell us like what microbes are present there and what functions were present and active in the experiments. And so this concept of functional genes, basically what it tells us is if this functional gene was active or present, it was potentially doing this function. So for methanogenesis, the functional gene that we could use to track this function would be the MCRA. And for the methylphosphonate assimilation or breakdown, we would use this fungi functional gene. And so we measure this along other different functional genes to try to figure out what was higher at the end of the experiments compared to the beginning and try to get some answers of what is happening where. And so here what I'm showing in this figure on the left is the transcript abundance, which basically means the quantity of the genes that we extracted from the RNA. So the higher of the abundance would mean there was more of them in the different samples. And we have different incubation experiments for the different panels. And there's like many different genes that we measure, but like for this, I just wanted to focus in the one that is highlighted here, which is the fungi gene that is gonna indicate that the methylphosphonate pathway was active. And we consistently find this to be active in the different experiments where we measure this. So this is an indication that this could be happening. And because these are oligotrophic lakes, it also would make sense that it happens since phosphorus is limited in this area and could be an alternative way which microbes are obtaining phosphorus in these lakes. In contrast to that one, we also measure the MCRA gene, which is shown here in this little box. And we could see that it's absent in some of the experiments and it's not extremely abundant in the different experiments. And what this suggests is that methanogenesis was probably not present in some of them, and where it had a very minor role in methane production. And so far I focus on the mechanisms of paradoxical methane production that are already known in the literature, that someone already kind of like measured and we know a way to track them. But I wanted to go beyond that and try to figure out, is there a mechanism that we're not familiar with that it could be present in these experiments? And I'm gonna try to walk you through the way that we went about to discovering this. And this is an example of one of the incubation experiments that we did, where you can see methane on the y-axis, methane concentrations, and the experiment over time. And on the bottom, this green line is the control, which means there was a lake water incubated, measured over time, but nothing was added on it. And the two bottom ones are the treatments, which is the methanogenesis inhibitor and the light intensity treatment. And the main takeaway from this figure is there seemed like to be a wide difference into the methane production that happened in the treatments compared to the control, which made it a good place to look for differences in the genomes or other type of techniques we can use within the experiment to see the differences in here. And so to further correlate like the difference between these treatments and the control, we can use stable isotopes measurements of the carbon from the methane gas samples to corroborate where the methane is coming from, and if it's from a different source. So we call that the isotopic methane carbon of methane ratio, and it can be used as a fingerprint of the gas in the source, which means that the ones that are more similar the ones that come from the same source will have the same or a very close isotopic signature. And if you have a different source, you probably have a significantly different methane signature. And so here what you can see is that this isotopic signature is lighter in these two treatments, and it's a little heavier on the control. And this is consistent with what we saw in the experiments, where the control significantly different to the other two treatments from the first time point, which is like the 25-hour time point. And what this indicates is like there's probably a different biological source in these methane treatments. And from looking at the literature, we found that this lighter signature of the methane isotopic signature is potentially related to cyanobacterial methane production. And so at this point, we knew something different was happening, and we wanted to look more into the genomes of if we could see this difference in the genomes as well. So we look at functions that were statistically significantly different in the treatments and the control, and specifically within the photosynthesis and carbon fixation area, since that's where we think the functions would lay out if it's related to cyanobacteria. And from looking at this data, we found two functions involved in the chlorophyll metabolism that again were significantly higher in the treatments versus the control. And we proposed that this could be a new potential mechanism of paradoxical methane production. And this is like these two genes called protochlorophylite and chlorophyllite, and DPOR and COR for short. And there's a couple of hypotheses of what we think is happening in terms of how the methane is being produced. And one of it is like, we have these big molecules that are these protochlorophyllite and chlorophyllite. And the ester methyl groups in these molecules could serve as precursors for methane under stress. So for example, this high light intensity could be producing some stress that generates methane as part of this process. And this has been observed in plants before, but not in microbes. The other potential source of methane from this genes is related to the fact that these two molecules these two genes are nitrogenized like enzymes. And this type of nitrogenized enzymes can catalyze reductions of multiband compounds into methane. So we propose these two mechanisms that could be happening within the context of these genes that we found. And we also found that these two genes are common in this, mainly in cyanobacteria, but also in like an important microbe found in lakes, which is called limnohabitant. And here I just wanted to show same experiment. You can see that the main genes that are higher in the treatments compared to the control are the ones involved in this new proposed mechanism compared to the other conventional methane cycling or the methylphosphonate metabolism. And just to validate further, we wanted to look, well, this happened in one experiment, but how about all the other experiments? And consistently, we found that these DPOR and COR genes are found across all the experiments and in different abundances and presence. So here I just wanted to, some of the summary and conclusions is that firstly, we observe the accumulation of accumulation of methane in aerobic incubations, which means that potentially aerobic or paradoxical methane production is active in high elevation lakes. We found that more than one mechanism can be producing methane under aerobic conditions in our incubations and potentially in the lakes. Methanogenesis, or the traditional process by which methane is produced, was unlikely the source of methane production in our incubations. But the expression of fungi transcripts suggests that methylphosphonate breakdown was involved in methane production in these lakes, as well as this previously undiscovered pathway that we propose during chlorophyll metabolism. And because it's like highly prevalent in cyanobacteria, could have potential implications in terms of cyanobacterial blooms and further climate change in the future. And that's it on my end. Thanks. I can take any questions if you have any. I think now is the
awesome. Thank you. That was a great talk. Yeah, so if anybody has questions for Dr. Perez or Professor Forrest, please drop them in the chat and I'll read them out now. So it looks like okay, it looks like Stephen had a question for Alex, which was, how much of the variation in flux estimates is related to the sampling and analysis versus actual spatial and temporal variability? And why would there be diurnal variability in the bottom sediments? Like, what drives the changes in sediments that are just sitting there in the dark?
So, Steve, the first part of the question, in terms of they are statistically different between sites, even though there is a big standard deviation at some of the sites that can be refined with taking more samples. Or like currently we're doing it 3 chambers for each location. We basically have a mooring site that we row out to, put out 3 chambers and then collect data during that time. If we had more chambers, it would help to refine that. But that said, that temporal variation absolutely exists, as does the spatial variation is not quite as convincing. And these measurements, to answer part B, are being taken from the surface. So what we're assuming is that the gas production rate is related to sort of a one-dimensional effect, that basically the water's not being moved around. And I think the diurnal variability is somehow linked to what Ellie was talking about in terms of the methane paradox at the deep chlorophyll maximum. And that's something that we haven't been able to capture yet because we haven't gone and done vertical profiles. We basically have measurements from the surface and we have measurements from the sediment, but trying to understand how the pathway the methane takes from the bottom to the surface is the next challenge, and trying to address that. Thanks.
Next, I had a question. So many reservoir owners and utilities have carbon neutrality goals. Do you guys think that greenhouse gas emissions from reservoirs make up a considerable fraction of total emissions?
Well, that's a loaded question, Mark, because that's the exact question we're trying to answer right now. And it really comes down to what's the footprint that you're trying to measure. And so, for example, a system like Clear Lake that is huge, trying to quantify that contribution will be much bigger than a system that's a series of smaller lakes or reservoirs. And so trying to upscale the measurements we've been making, both across Valley Water and then across different microclimates around the state, I think is the next step. And so if anybody's interested in partnering with this, we're trying to collect more data across a range of different trophic states, a range of different latitudes, and a range of different climates. Sorry, not altitudes, and a range of different climates. So we can start grouping water bodies to understand what the total contribution to greenhouse gases is for the state. But that's something that we just haven't quite got far enough along yet.
Thanks. So it looks like Alex Horn has a comment. Alex says, it's likely that the sediment emissions in summer are a pile of sunken algae. I use this hypothesis to explain nitrification of ammonia to nitrate and subsequent denitrification as the water slowly rotates around the bottom.
So I would agree, but before I answer that, if you can jump to the next question by Dendi.
Cool, yep. So it looks like, yeah, so have you evaluated the organic matter content in sediments with flux chambers? Since methanogenesis is a terminal process in organic matter degradation, we would expect organic matter to be high in sites with high diffusive or emulative flux.
And the short answer is yes, and we were disappointed by the results. So we gathered sediment samples from multiple locations around the lake to see if we could see a correlation between that and the high gas volume that we were seeing in the acoustic response. And we're not convinced yet. We're going out and gathering more samples. And so I actually like Alex's hypothesis more of the sunken algae idea that's somehow contributing rather than the sediment itself. But this is something that we are actively exploring. The LOI results we just got back about 3 weeks ago. And then we're also looking at pore water content and DOC to try to understand other potential mechanisms that are happening at the sediment water interface.
Thank you. Next one's from Professor Butel for Dr. Perez. The bacteria you identified as potentially producing methane under oxic conditions with newly proposed mechanism, what type of bacteria is this? What metabolism does it use? Aerobic respiration? What conditions do it thrive on?
Yeah, so we found this mainly in cyanobacteria, which wasn't extremely abundant in the oligotrophic lakes that we had, but it was mostly cyanobacteria. And then there was this one type of protobacteria that is called limnohabitans, and it produces, it also has the capability of making photosynthesis, but it's like an oxygenic photosynthesis. So I think the main kind of like consistent way that this happens is during photosynthesis while they're producing the chlorophyll, which means like it could potentially have implications about this. And I mean, this is also being explored by other research groups under different conditions. And it does seem like cyanobacteria could be very important in methane production. Yeah. But yeah, so we're still kind of like learning more about this specific process. Yeah.
All right. Looks like Mark has another question for Alex. For characteristics that correlate with methane production, you showed area. Was that surface area? What about watershed area, which could correlate with external nutrient loading and allochthonous carbon loading?
Yes. I'll just stop. No, that was from the Deemer's paper, and I believe it's surface area, that initial plot in terms of the literature review. I agree with you that watershed area plays a role. And then the other thing that plays a role I'm hypothesizing plays a role is wildfire. That years that a wildfire sweeps through a basin, then the runoff from that year will be much higher in terms of sediment loading. And we know this, there's a lot more sediment loading, there's a lot more potential phosphorus loading coming into a system. And so that's based on watershed, what's coming in from just sort of baseline conditions, and then when you get these wildfires come in. So I think there is a characteristic response that will be embedded in each lake system that you look at. And I don't think we have the answer for that yet. Although I think understanding the baseline without fire or without anything else is the first step.
Thanks. Yeah, so if anyone else has more questions, drop them in the chat. I have another one actually for Dr. Perez, which is, so I've noticed one of the things you mentioned earlier was that one of the hypotheses for the methane paradox was potentially like zones of low oxygen related to like suspended material in the thermocline. So like potentially like algae cells or inorganic particles or something. And that kind of seems similar to an issue we deal with mercury, which is water column methylmercury production. And that's kind of postulated to be, you know, basically due to that same mechanism. And so I can see some parallels there. And I was wondering, in your research, have you kind of ruled that out as a potential mechanism, or do you still think that that could be playing a role?
Yeah, so for the specific incubations, I don't think that's ruled out, and it probably does happen in lakes to an extent. For our incubations, we filtered some of the particles, so that wouldn't be a role in particular in ours, and because there wasn't that much methanogenesis either, it doesn't seem likely, but I think that is definitely a possibility within lakes that have a lot of detrital like inputs, yeah.
Gotcha. So you basically like filtered them enough to remove big particulates, but not enough to remove bacteria?
Yeah, basically, yeah.
Okay, gotcha. Thanks. Looks like Alex had another comment. Lumps of cyanobacteria are often common, if not often noticed, in high oligotrophic lakes. Could this be important for methane production?
Yeah, so we do have some of this in our data. I would say that in the lakes that we were studying, protobacteria was the majority, or at least in the incubation like filters, protobacteria seems to be the majority of the bacteria present. Cyanobacteria were present to a lesser extent, and I don't know, it could be just that the protobacteria were some of the ones that we had in our samples, kind of like relay on decomposed organic matter, so that might be there longer maybe than the cyanobacteria that might be just run out of nutrients at the beginning of the incubation and not make it, I guess, to the end of the incubation. But yeah, it's potential, like we did see it, but not as much as these other protobacteria.
Any more questions for the speakers? If not, I'm just going to give another quick little shout out. So we do have a California Lake Management Society store, and I just got rid of it. Here it is. And so yeah, you can go on to I think it's like Cafe. It's linked on our website, california-lakes.info, something like that, or sorry, .org. And yeah, you can go on and get a conference hoodie or a pin or a cup, and we're also going to be giving a small gift to each of our speakers. So yeah, thanks for checking that out. And just another reminder, we have another session tomorrow. It starts at noon. We're going to have our 4 student scholarship awardees. They're going to be giving kind of like little lightning fast talks. And then we have our keynote address, which I highly recommend that you tune on for. And in the meanwhile, if the speakers could drop their contact email into the chat, feel free to follow up with them. And yeah, thanks everyone for joining.
Okay, Mark, thank you very much. I'm going to sign off. Ellie,
Yeah.
thank you for a great talk.
Awesome. Yeah, thank you.